Watershed. Uh, and the Oregon Community Rights Network has a project with called Water Is Us, and this is a part of that. And it's assisting in Lane County's initiative effort to get better protection for watersheds in Lane County. Anyway, with that, I'll turn it over to Jed and Amanda. Thank you for coming.
You bet. Yeah, thanks for the opportunity. Yeah. So we're going to kind of team up on the presentation and each kind of jump back and forth a little bit as we work through it, and definitely encourage anybody just to, fire away with questions if you have them. Just yeah, speak up or try to raise your hand through Zoom or whatever works best for you. Yeah, I'm Jed Call. I'm a fish biologist, and I've worked at the Long Tom Watershed Council for 16 years now, mostly focused on aquatic habitat restoration projects is kind of my main duties here. So yeah, working with lots of landowners to develop a wide variety of projects that I'll talk about as we work our way through the presentation. Um, yeah, so I'm just going to start with like an overview of the watershed and of the Long Tom Watershed Council. It sounds like you guys did a really good job kind of with the overview of the Long Tom Watershed and the presentation that David mentioned. But just a quick review here. This is a map of the Long Tom Watershed and the Long Tom Watershed Council service area. They're two separate geographies, kind of. The Long Tom Watershed is 410 square miles. It's a little bit unique compared to like the Siuslaw or the McKenzie in that it is 92% privately owned. So yeah, lots of agriculture in the kind of valley floor, lots of small woodland owners and industrial timber in the kind of fringes in the Coast Range and in the southern part of the Coyote Creek and Spencer Creek watersheds. Lots of rural residential uses and a handful of small communities. And then, of course, Eugene and Springfield and urban areas. So it's pretty diverse for sure. This map shows the different sub-watersheds within the Long Tom watershed. We'll be talking about a few of those more than others as we go through the slides. Bear and Ferguson Creek are just west of Junction City coming out of the Coast Range. Can you see the cursor there moving around?
Yeah.
All right. Perfect. So yeah, I'll be talking about those areas when I talk about some of the habitat restoration work that we've done. And yeah, Amanda's got some monitoring results from Amazon and other parts of the watershed as well. And you can see that, yeah, our service area is slightly larger than the Long Tom watershed. We work with landowners along the main stem Willamette from Eugene all the way north past the confluence of the Long Tom and the Willamette. And we also work collaboratively with the McKenzie and Middle Fork Willamette watershed councils in the Eugene Springfield urban area on urban stormwater improvement projects. Lots of words on this slide. I think it's the wordiest one we have, so we'll get through it right at the beginning and be done with the words. But here's our mission statement. The Long Tom Watershed Council serves to improve water quality and watershed condition in the Long Tom River Basin and surrounding drainages through education and collaboration among all interests, using the collective wisdom and voluntary action of our community members. Watershed councils are a little bit unique. You guys probably are familiar. Oregon is, there's lots of watershed councils in Oregon. I believe that almost every watershed has a council now. A lot of support through the state government, through the Oregon Watershed Enhancement Board. Lottery revenues help fund us. Most watershed councils started in the mid to late 90s, as far as I know, when the state government set up kind of a formal structure for them and a funding mechanism through the Oregon Watershed Enhancement Board. We're a little bit unique in that we are, I think, bound by statute to work with all interests throughout the watershed, from people that are interested in water quality and habitat conservation, to farmers, to foresters, to urban residents, to scientists. Everybody gets a seat at the table. So, and the goal is to work with all of those different constituents and hopefully work with them to make improvements in the way that they manage their land, wherever they're at in the spectrum. So the Long Tom Watershed Council, we started in 1997. First employee started in 98, became a IRS nonprofit in 2007. We're mostly funded by competitive grants, almost exclusively. We have a board of directors that represents all of the diverse interests from throughout the service area. We have three main program areas. Rural habitats, that's mostly where Amanda and I work. Amanda also works with the urban program as well. Work is very diverse: water quality monitoring, habitat restoration projects. We have coworkers that work on prescribed fire and oak and prairie restoration. We have coworkers that work in the urban environment to improve water quality, like I mentioned. And we also have a traditional ecological inquiry program that focuses on working with tribal youth and their families to help them explore projects focused on indigenous land management practices. So lots of action going on. I encourage you to check out our website and learn more about those other things. We're mostly going to talk about water quality monitoring and aquatic habitat restoration projects today. So just a small kind of subset of the work that the council does. I think it's Amanda's turn. Yeah.
All right. My name's Amanda Reynolds, and I've been with the Watershed Council for, I think, about 8 years. And I'm going to focus exclusively in my portion of the presentation on water quality monitoring. And for the majority of that, I'm going to focus on what we've been doing in the past like decade-ish or 8 years, kind of since I've been part of the Watershed Council, and the more recent, more current data we have available. But I'll spend, if we have time, a little bit of time towards the end, kind of summarizing what else there is and where else you might be able to go to get more information outside of the data that we've collected as a Watershed Council too, for anyone who's interested. And the work that we've done over the past 8 or so years has been primarily collecting water quality data on pesticide presence in water, stream temperature, and E. coli bacteria. So three main water quality initiatives in recent history. And I'm going to start with pesticides. We've been doing pesticide monitoring in Amazon Creek and only Amazon Creek since about 2011 as part of the Pesticide Stewardship Partnership Program, which is an Oregon Department of Agriculture, an ODA program that's run in partnership with the Department of Environmental Quality. It's a totally non-regulatory monitoring program that focuses on voluntary changes in response to identified pesticide problems based on the data we're collecting. And in Amazon Creek, so we've got our map of Amazon Creek, we are taking grab samples, we're dipping bottles in the creek about once every couple weeks throughout the spring, summer, and fall, and sending them to the DEQ lab for an analysis. They analyze for a suite of about 135 different pesticides. And we've been taking these samples at 5 locations in and around the city of Eugene in Amazon Creek to get kind of a variety of different land use signatures. So we've got pretty residential signature, the sampling sites near the 29th Street dog park. We've got a site out by Beltline Road on West 11th that captures more commercial activity. We have a site capturing some industrial activity on the A1 channel off Aubrey Lane, kind of west of 99, east of 99, sorry, still my directions backwards sometimes. And we have our most downstream site captures more of an agricultural land use signature. There's a lot of grass seed farming, some other mixed agriculture in the southern part of Amazon Creek that we're able to kind of get a signature for.
Real quick, do you guys put all that up on your website, that data?
Not on our website. Generally, there is a lovely tool that the DEQ posts for public use, the PSP data viewer. The DEQ experienced a data breach, a hack, like 3 months ago that brought down a lot of their data services, and this one hasn't gone back up yet. It's a priority for people working in the PSP at the DEQ, but it's kind of not up to them. There's a larger cybersecurity barrier at the moment, but I'm hoping it'll be back up because it's
Okay.
really, really nice tool for viewing the data and viewing it like in the proper context too. They've got some good tools that give some contextual information about what you're seeing.
Right. Okay. Thank you.
So I did not include a link because it's not live, but it should be again in the future. So we would get this data where they're analyzing for like 135 analytes. So we get back what analytes are seen, which pesticides or pesticide breakdown products are seen, and at what concentrations. And to make sense of it, we have to compare it to something. And there aren't really water quality standards for pesticides, but the EPA does publish aquatic life benchmarks, which are thresholds in the concentration of different pesticides where, according to the best science available, we know it's probably going to harm something in the aquatic system. So for any given pesticide, there might be like 5 different aquatic life benchmarks that are relevant to fish or macroinvertebrates or plant life. And the program takes the lowest of those, so the most sensitive threshold, and compares the concentrations of that pesticide at that kind of lowest level value at which we think it might do harm to something in the aquatic system. Just to get more of an apples to apples comparison, like what does this concentration of pesticide mean in the waterway in a more realistic sense. We're also looking at how often we're seeing any pesticide in the water. Is it very rare? Are we seeing it all the time? And just overall kind of the number of different pesticides we're seeing in a given sample or in a given year. We're looking out for the kind of cumulative and synergistic impacts that are a little harder to know, but are still worrisome when we're seeing pesticides in water. And this is a quick snapshot of pesticide results over the past 3 years, which is kind of the window during that we use to determine what pesticides are of concern. And the vast majority of pesticides, so we're seeing 24 different pesticides or pesticide breakdown products over the past 3 years of that suite of like 135. And most of them, all the ones in this kind of greenish box, are products that we're seeing rarely and at concentrations that are well below the aquatic life benchmark. So we're seeing them, and that's of some concern, but generally they're at low enough concentrations and we're seeing them infrequently enough that we're less concerned about this suite of pesticides. There's another set of pesticides that we consider of moderate concern because we're seeing them very frequently, sometimes almost 100% of the time in our water samples, but at very low concentrations compared to the aquatic life benchmark. And that includes Roundup, glyphosate, the active ingredient in Roundup, and its breakdown product, ANPA, as well as a breakdown product of another really commonly used herbicide, diclofenac. The breakdown product is sometimes called VAM. So we're seeing them often, but not at levels in a given sample that we know would do harm to any constituent in the aquatic environment. There's another set of pesticides that we're seeing occasionally at higher levels. Notably in here, I think for especially folks in the urban environment, is imidacloprid. It's an insecticide that's used in like 250 registered products. And we see it randomly all over the urban environment and very rarely in industrial and agricultural areas. And pretty much whenever we see it, it's over the aquatic life benchmark because it's very toxic at very low levels. It does a good job at harming insects and tends to harm our aquatic macroinvertebrates. And then what we consider a pesticide of highest concern, diuron, we see fairly frequently and often at levels that are approaching the aquatic life benchmark or are slightly above the aquatic life benchmark. And we're seeing that more in agricultural areas. So we're seeing that farther down in the watershed. Very quickly, I can't spend too much time on any one thing, unfortunately, but I wanted to give you some sense of the fact that we are seeing these pesticides everywhere. Naturally, as we're going downstream, you would expect to see more and more pesticides as they're kind of like attenuating with water flow downstream. So we are seeing that, and that's kind of hard to parse out from like new, that signature is a little hard to separate from, like what's coming in from further down in the watershed. But we see exceedances everywhere in rural residential, in agricultural and industrial. And we tend to see more of these exceedances in the industrial and furthest downstream our agricultural site, but it's not exclusive to those sites. And then I wanted to, this is really hard to read, I'm sorry, but I want to remind myself to mention that overall, over the past 15 years, we have seen some positive trends in our data results. So there's a column here for our PSP, our Amazon Creek PSP, and the green squares mean things that are getting better. So things, each analyte on the left, each of these pesticides for which there is a green square is one for which we're seeing less and less of it over time. This data is a little bit dated, but we are still seeing pesticides dropping off of our 3-year list, and generally seeing, with some exceptions, positive trends in our pesticide results. They're not huge, major improvements, but good slight forward progress over the years. So what are we doing as a watershed council to address pesticides? We promote integrated pest management, which can include not using pesticides, or can include minimizing use of pesticides, using them responsively and responsibly. We host integrated pest management workshops for pesticide applicators, something we've been doing recently. And we also promote a lot of best management practices that can help attenuate or capture pesticides that are moving into the environment in urban areas like stormwater management facilities, or in our rural areas, riparian buffers, filter strips, grass waterways, different things that can help capture sediment and pesticides before they're moving into our streams. And now I'm going to jump to stream temperature. So we've also been measuring stream temperature pretty consistently, actually, over the past 15 or so years, but I'm going to focus on a little more recent data. We capture hourly stream temperatures throughout the summer. So we've got little data loggers that are living in the streams. And we've only been doing this in Bear and Ferguson creeks. So we don't have City of Eugene data. Probably obvious to some of you, but I want to briefly mention a few of the reasons that we're worried about stream temperature. We've got native fish and other aquatic organisms that are quite sensitive to high temperatures. And high temperatures can also contribute to other water quality problems, notably algal blooms and cyanotoxin blue-green algae blooms that can further degrade water quality and cause issues for drinking water providers, like the city of Monroe that pulls its drinking water from the Long Tom downstream. Things that can contribute to high stream temperatures, direct solar input. The shortwave radiation directly from the sun is probably the most obvious, but there's also kind of that long wave ambient heat that's harder to do anything about. And water quantity, I think, is the less obvious contributor to high stream temperatures, because water is difficult to heat. The more water you have in the stream, the cooler it tends to stay. If there's less water, it's easier to heat, and so it tends to get hotter. Here quickly is a snapshot of our recent temperature monitoring. We've got our Ferguson Creek watershed here, our Bear Creek watershed here, so City of Eugene's down here, Amazon Creek, Long Tom River. And we're taking our hourly stream temperatures, we're getting daily maximum temperature values and getting a 7-day average moving window value for that. So we're looking at kind of daily high temperatures and comparing that again, so we can make sense of this data, like how hot is too hot. We're comparing it against state standards. In this case, a standard of 18 degrees Celsius, kind of like a temperature that's comfy for our native fish. Below 18 degrees, they're pretty happy. And you can see, so most of our watershed, all these oranges and reds, were exceeding 18 degrees either for like a month to most of the summer. A few places higher in the watershed, and we're still monitoring these lower reaches of the watershed because we know they're hotter. So I should add there's cooler temperatures up in our headwaters. But further down, temperatures are getting uncomfortable for native fish throughout the summer. But I also want to go back and look a little further back at some of the older temperature data. And I'm sorry, this is really hard to read again. But this is data from a watershed, a really comprehensive watershed assessment that I'll refer to again later, that was done about 20 years ago. And notably, here I only have a single criteria of days over 18 degrees. In this older data set, you will see streams that are symbolized for exceeding 22 degrees, which is considered like a lethal standard for a native fish for at least 4 days of the summer. And there were a lot of lower segments of streams that were meeting that criteria, including lower Ferguson Creek and lower Bear Creek. And we didn't see that in more recent monitoring data. This is not like a statistically valid trend analysis, but a nice positive indication that it might be getting better. We are seeing not as many lethal temperatures in recent data. So that's pretty cool. What are we doing, speaking of cool, to keep streams cool? A lot of planting trees and shrubs along streams to prevent that direct solar, like shortwave radiation. Our friends at the Upper Willamette Working Lands Program, Upper Willamette Soil and Water Conservation District, NRCS especially, help farmers with irrigation efficiency projects. We make referrals to those programs where we can to keep more water in the stream, to help keep streams cool, to address that low water criteria for heating streams. Another kind of sneaky source of heat in streams can be these like in-stream reservoirs that sit and bake in the sun and then have surface spillways where we're conveying just the hottest water from that in-stream pond downstream. There's some complexity to that. It depends on the specifics of each case. But dam removal and modification in some of those structures can have big impacts, positive impacts on water temperatures. That's something that type of project that we work on with some frequency. Also, wetlands, beavers, encouraging groundwater interaction can decrease the temperatures of streams. Again, some complexity there. But it's good for a lot of other reasons, so we do it for other reasons as well. And finally, our final set of recent water quality monitoring data is E. coli, which again, we're getting grab samples for E. coli, getting scoops of water. We did this over 2022 to 2023, so kind of a shorter term study, sent those samples off to the lab. Why did we do that? Why are we getting E. coli counts? There's actually nothing in particular about E. coli that we're too concerned about. Some E. coli is fine, some is not good for you, but it just happens to be a really handy thing to count. And it's a good proxy for fecal contamination. So there's E. coli, it means there's poop getting in the water. And there are other things that come along with that, other nasty microorganisms, as well as nutrient pollution that comes along. So when you see E. coli counts, you can think just like fecal contamination in general is what we're thinking about. Where does it come from? The guts of animals, and those animals can be our livestock, our pigs, our sheep, horses are also a big one. It can come from our guts, frequently our septic systems, sometimes municipal wastewater systems, and also wildlife, which is harder to do anything about, but can be a big contributor to E. coli in places. So again, we're getting these E. coli counts back, which happen to be like counts of organisms per milliliter of water. How do we make sense of that? We're going to compare it against a water quality standard for some idea of how much is a problem. And in this case, we're looking at an acute water quality standard. So a single sample with a level that exceeds about 400 organisms per microliter is considered above the standard. And how often are we seeing results that are above the standard? In some cases, like here in Lower Amazon Creek, and also a site in Bear Creek, in the Owens Creek, tributary to Bear Creek, over 40% of the time, we were seeing exceedances of that water quality sample. Quite a bit. Lower portions of Barron Ferguson Creek, 15 to 40%. And then a few places higher up, we were seeing fewer exceedances. Again, we chose these sites because we expected to see high values. So we would expect to see much, much lower E. coli counts higher up in the watershed, or potentially other places in the watershed. But we were looking further down where we expected we might see more problems. Interestingly, Amazon Creek, just outside of the urban growth boundary, had very little E. coli, which I would not have guessed. Again, I wanted to kind of compare to how this looked 20 years ago, and this is a little more of a mixed bag, but you can see this, again, the symbology is different, but we're using the same break values. So you can see this lower Ferguson Creek and lower Bear Creek in the older data exceeded the criteria over 40% of the time. It was 15 to 39%. So those sites were better during the time period that we were sampling more recently. There's some improvement in the E. coli counts. A few other sites like lower Amazon Creek and that one site in Owens Creek were, looking worse in our more recent data. So a little bit of a mixed bag, kind of hard to parse out, but some improvement, some declines. It's worth saying that E. coli changes a lot through time, both during a day, throughout the year. It's kind of, it's a parameter it's a little harder to kind of get a good hold on. And the patterns that we were seeing in the data indicate that it was coming from everywhere. So we saw some sites that had result patterns that were a little more stable through time, a little higher in the wetter season. They're indicative of septic system failures and septic system inputs, and then some sites that were almost certainly the result of livestock. So people and animals, livestock, both to blame. What are we doing to help keep fecal matter out of the streams? We work with great folks at OSU Extension, if you're interested, that have wonderful resources on septic systems and septic system maintenance. We do some fencing work, but again, agricultural best management practices. We work with our friends at the Upper Willamette Working Lands Program and the Upper Willamette Soil and Water Conservation District, particularly, can help with some of those projects. So just to kind of summarize some of that recent water quality monitoring, we have seen impairments in each of those three areas in pesticides, water temperature, and E. coli. We have strong evidence that these impairments are coming from all sectors: urban residential, rural residential, agricultural, industrial, kind of everywhere is part of the problem. And some statistically valid evidence that things are improving over time, and some kind of like qualitative evidence with some of the other criteria that we might be seeing some improvements through time, but there's some mixed signal in there too. And then I know you are all very interested in water quality, and that was a pretty like narrow view of water quality in the Long Tom Watershed. So I just really quickly wanted to talk about some other places that you could perhaps go to see more data if you are very interested, as well as kind of preview something that we'll be talking about. That old data set that I referenced, the 20-year-old data set, Cindy Feynman, from 1999 to 2006, led a wonderful, really comprehensive, more geographically diverse assessment of water quality, and created this lovely report that is very accessible in terms of like readability and understandability. It's on our website. It is hard to find on our website, but it is there. I have a link to it here, so if you're able to like share out the presentation, you can follow the link. And I highly recommend checking that out if you want to look more into water quality in the Long Tom. It's dated, but it's great. We've done some other cool studies that Jed's going to be talking more about in his slides. A culvert inventory. All these purple dots and all these sub-watersheds are culverts that we've inventoried and assessed for fish passage and prioritized for replacement. So a really cool data set that we've used really heavily in projects. There's a really cool cutthroat trout monitoring study where hundreds of fish, maybe more, were tagged and tracked to see where they are and where they're going. There's going to be a paper out actually soon about that, but Jed will talk a little bit more. And these green dots here are from a rapid bioassessment that we've also used to inform projects, and Jed will be talking about that a little more too. The city of Eugene does annual water quality monitoring, and it is hidden very well. It's part of their municipal stormwater permit requirements to monitor water quality in waterways in the city. And they report on it every year in their annual stormwater permit. I'm including a link on this slide, but they've got great data and graphs. Notably, zinc is something that keeps coming up as analyte that's getting worse over time. Very toxic to fish. They're seeing it everywhere. It's something that we've never done monitoring for. So for more on that, you can try to either follow the link on our presentation or Google the Municipal Stormwater Permit, City of Eugene Municipal Stormwater Report. We'll get you there. DEQ has one long-term monitoring site. I've included some of their data in what I showed you, but there is temperature, E. coli, and turbidity data available for a single site on Amazon Creek, long-term data through the DEQ, but there isn't a lot of long-term monitoring done by agencies in the Long Tom Watershed. And finally, I know folks have brought up cyanobacteria, the blue-green algae. So I wanted to briefly touch on them and the resources that are available. Unfortunately, there's not a lot. So blue-green algae are not true algae. They're like a bacteria that is naturally present in waterways but thrives in warm, nutrient-rich water. And they can produce cyanotoxins that are harmful or even potentially deadly to animals like dogs or people. The Oregon Health Authority issues advisories when they know about cyanotoxin blooms. So that is a resource, but I think it's based on someone observing and reporting that. Jed just noted that there is a new advisory for Fern Ridge Reservoir right now that that will be investigated and either confirmed or removed. But that's the best like actual in-person monitoring source. But there's also an EPA tool called CYAN for Cyanobacteria Assessment Network. And you have to create like a little login, but it is a remote sensing tool that estimates the amount of cyanobacteria in larger water bodies. So it includes like Durana Lake, Triangle Lake, Fern Ridge Reservoir, and can give some indication, although it can be skewed, about whether and how much cyanobacteria are in our water body at a given time. So wanted to bring up those tools just because there's been recent questions and recent interest. And I think with that, I'm going to end my presentation and get off to Jed. Unless anyone wants to ask questions first about water quality. I know there's probably like kind of a tease, but
yeah, I'll transition to talking about habitat restoration projects and how we've incorporated some of that data. So if anybody does have specific monitoring questions, it would be a good time to pop those.
I know there's like super quick views on the results, so I'm sorry for that, but I wanted to kind of.
Yeah, no, great information because we're looking to try to test some water coming up here, and so I'm gonna definitely be taking a look at your data. That's great.
I have a question.
Okay.
Can we go back to the graph that had red, green, yellow, and a data set that was quite complex?
The pesticide one.
Oh,
Keep going.
The rectangle.
The pesticide, this one? There's a lot of red and green.
Yes.
And yellow.
Yeah, that one. That one. So you mentioned that the green indicates some improvement in those particular chemicals. What is the yellow and the red indicate?
Yellow means there's no statistically valid trend for the assessed period, which is a little dated at this point. I think this was like done in 2020 or 2021. So looking at the first like 10 years of data from the PSP, I haven't seen a newer one out. And the red means an increasing trend in the concentrations of
Okay, so
that.
in the yellow, it doesn't mean that it's not there, but it's there in a small way.
The yellow means that this particular graph isn't really related to how much we're seeing. It's just whether we're seeing more or less of that thing. So
Okay.
gray means we're not seeing it at all. These aren't pesticides that we have been seeing in the Amazon Creek, which is great. They're not relevant to Amazon Creek. Yellow means we're seeing that things, in this case, atrazine, probably about the same amount over that 10-year period. There hasn't been a notable increase in like the frequency or concentration that we've been seeing atrazine, or a notable decrease.
And it confirms that it's a forever chemical, isn't it?
And then, yeah, red means we were seeing more of it over the course of that 10-year period, which is sulfamethyl, methyl, and glyphosate in this particular window. And then all the green are things that we were seeing less of over time.
Okay,
Which
thank you.
interestingly include the breakdown product of glyphosate. So we're seeing more glyphosate and less of the breakdown product of glyphosate. So some of it's, yeah.
Thank you.
Yep. Anyone else have a question before we move on to some of the projects, some of what we've been doing with the data?
Guess
If
not.
nobody else has a question, I've got another one.
Okay, go for it.
On your maps of the places where there are higher temperatures and more pollutants and that sort of thing in the watershed,
Mhm.
is there any correlation about where industry is in this area? Because I'm not familiar with this watershed particularly, so I'm just curious if there's anything that stands out as a reason to explain some of that.
Yeah, and so for these temperatures, the more recent temperature data, that's a great question. And Barron Ferguson Creek, all of these monitoring sites are in agricultural areas.
Okay.
And further downstream, there are stretches of the creek that lack adequate riparian vegetation. And there's some conflict, I think, specifically in Turnbow Creek, there's also some like in-stream reservoirs and things like that I think are causing interesting temperature signatures. But a lot of it is agricultural land use where we're lacking riparian shade.
Okay.
Although we don't have recent temperature data of this quality for Amazon Creek. But Amazon Creek is a good case in point that urban areas contribute a whole heck of a lot of heat to the creek too. Grab temperature, so just like kind of single sporadic temperature samples in Amazon Creek outside of the urban growth boundary have revealed some really high temperatures, like above 20, 22 degrees. That do increase downstream because it goes through an agriculture area. Lacks adequate cover also.
Yeah.
But the high temperatures are definitely coming from our urban areas too. The concrete channels, lack of shade in place are big contributors to high water temperatures too.
Thank you.
You mentioned that 92% of the Long Tom Watershed is privately owned, and with the forest land, do you have a breakdown of the forest land within the Long Tom Watershed? How much of it is industrial forest land um, that, is scheduled for, 40-year rotations, that kind of thing. Versus there is some BLM up in the headwaters, I know, and
Yep, there's Bureau of Land Management, burst land that is used for timber production. There is large industrial timberland, like Weyerhaeuser, and then there's also small timber holders,
yeah.
small woodland owners. And I forget, I think I do have some analysis of the
Yeah.
mix. It was like almost 50-50 between the large holders. Does that sound right? Like
I'm not
smaller
sure. Yeah,
timber.
we
I don't have that
yeah.
on hand.
Yeah, that data does exist, and we can follow up with that if you're curious. I think it's probably part of another kind of foundational document that we have on the website, this watershed assessment that looks at all sorts of different land uses and geological history and stream modifications. So yeah, the Long Tom Watershed Assessment does have some land use breakdowns that I believe go into that level of detail.
And there's a significant amount of all of those things.
Yeah. Thank you. That'd be great. I'd like to see further breakdown, but no rush to get it.
Cool. Well, maybe I should jump into my slides. It'll take me about probably 15 minutes. So we'd be pushing 7 o'clock. I don't know if is that all right with everybody or?
Sure.
Okay.
Yeah, I think so.
Yeah.
Okay. How do I get this going
So then you can drop down and do a presenter view. You've got note too.
Can you guys see this slide with the map on it now?
Yep.
Yes.
All right. So what I'm going to cover is mostly focused on the on-the-ground work that we've done with landowners throughout the watershed, with a little bit of a focus on those same sub-watersheds, Bear and Ferguson Creek, west of Junction City. And I just wanted to first point out that on our website, we have an interactive projects map that you can cruise around the watershed and click on all those colored dots on the map that represent projects that we've done or are in the works. And there's photos of some of the projects. There's more information. It's kind of just an overview of each one of those, but I really encourage you to check that out on our website, and you can see all the things that we've done and what we're up to on a map like that.
It actually is easy to find on our website, unlike some of the other stuff.
Yeah, I think it's right on the homepage. It should be.
A link.
Yep. So just wanted to, you've heard Amanda talk a lot about Bear and Ferguson Creek, and I just wanted to briefly touch on why that's important from a species and river perspective. Bear and Ferguson Creek, those two subwatersheds are located downstream of Fern Ridge Dam, and that's what's on the slide there. Fern Ridge Dam has no fish passage facilities at all, so it's a total barrier for aquatic organisms trying to migrate upstream. It blocks 2/3 of the watershed that was historically accessible for migratory fish. Fish species that are of particular interest for us are coastal cutthroat trout on the top here. Those fish I'll talk a lot about them as we progress here, but they can move long distances depending on their life history, looking for different habitats. So Fern Ridge Dam obviously impacts them pretty significantly. Second fish here is a juvenile spring Chinook. They're present below Monroe, and I'll talk more about them as we move along here. And then this is a Pacific lamprey that historically used the Long Tom Watershed as spawning habitat. They're anadromous, just like salmon, prehistoric fish. They are still present below Fern Ridge Dam in small numbers in Ferguson and Bear Creek. They're a culturally important species for indigenous people in the Northwest as a food source. So they're another priority species for us. So I'm going to kind of talk through some of the data that we've collected. Super quick overview. Amanda kind of gave a plug for some of these little studies that we've done over the years, and I'll just go into a little bit more detail and talk about how we've incorporated what we've learned in those studies. From 99 to 2006, we as part of that comprehensive water quality monitoring effort, we collected macroinvertebrate samples throughout the watershed. They're an indicator of water quality and stream health. So some of the kind of conclusions that we came to after that part of that study were that streams with dense native riparian vegetation and good shade had higher macroinvertebrate diversity and abundance. Similarly, sites that had more in-stream wood, like log jams, had slightly higher macroinvertebrate scores. The culvert surveys that Amanda mentioned, that was one of the first things that I did when I started at the Watershed Council in 2009. We had 4 seasonal workers that went out and surveyed almost 300 sites throughout the watershed. Almost 80 of those were in Bear and Ferguson Creek subwatersheds. It's a big project, lots of landowner outreach involved. The results of that indicated that about 82% of those crossings did not meet state and federal fish passage criteria. So there were barriers for juvenile trout passage. So that's a lot of fish passage barriers, 82% of 300. So we don't have the resources to replace all those with fish-friendly crossings. So we worked with scientists to prioritize streams for fish passage projects, and that was kind of based on habitat quality. The goal was to remove consecutive barriers to be able to allow these migratory species to get into important habitats for spawning or for cold water refuge in the summer. Another one of the pieces of data that we have collected and used to help prioritize on-the-ground projects is the rapid bioassessment snorkel surveys that Amanda mentioned. We looked at 4 different streams in the early 2010s. I think it was like maybe 2010 through 2013 or 14. Got to get in the creek, and we counted all the fish in every 5th pool in those streams. A lot of this is a pretty common survey protocol that's been used in the Siuslaw watershed a great deal, and many other watersheds throughout Oregon. And basically, you just count the fish in every 5th pool, and you get kind of a density of fish per square meter. And that can give you an indication of where the fish want to be, and theoretically where the high quality habitat is. One of the main findings from that is that cutthroat trout density increased as you moved upstream. And that correlates with where we were finding cooler water temperatures in general and more spawning habitat. They need gravel to spawn, and more intact riparian areas as well. So a lot of things kind of made sense in terms of connecting the dots. Another piece of information that we've used to help prioritize projects was tracking the movement of cutthroat trout with a ton of help from volunteers. Thousands of volunteer hours were involved, including Amanda before she was on staff here. We collected over 1,000 trout. We tagged 600 of them with pit tags. Those are similar to the tags that go in like your pet dog, with your contact information. It's the same technology. It's passive integrated transponder is what PIT stands for. Some of the findings from that study just basically indicate that cutthroat, and we kind of knew this going into it, they have a wide range of life histories. Some can live in a small section of stream for their whole life, maybe only a couple hundred meters. But on the other side of the spectrum, some move great distances throughout the course of their life. So I think the big finding that was interesting for us was that about 35% of fish that we tagged moved at least a half mile, and over 20% moved more than 5 miles. So that tells us that a good proportion of these fish, in order to express their life history and find a suitable habitat, they need to be able to move long distances around the watershed, which further helped us prioritize replacing fish passage barriers in certain streams with good habitat. Another finding from that study, though, is that there are some outlier fish that can make it over significant barriers. It's really amazing to see what they were able to do. The photo on the left is one of 3 Army Corps lowhead dams on the lower Long Tom that I'll talk more about. Thought to be a total barrier for cutthroat trout. Our study proved that they can get over those 2 pieces of the dam there under certain flows. And then also this culvert on the right image has about a 6-foot drop at its outlet. And we were able to show that Cutthroat could get around that. And the secret to this is that it's during really narrow windows of flows, like during floods, for instance, that culvert, fish could, use the floodplain to get around that culvert during big floods. Similarly, on the Lowhead Dam there, during a real narrow range of flows, fish could jump over those two vertical barriers there. So it's still really important to replace barriers like this and do what we can to improve fish passage, because they are slowed down in their migrations as they wait for the right flows to emerge, and they can be preyed on by other animals or be subjected to poor water quality in some cases. So not saying that we shouldn't improve fish passage, but it was just a really neat finding that we had from that study. So kind of just a quick summary. What have we done with all this data? It helped us prioritize projects that improve riparian shading and native tree and shrub cover along streams. It helped us prioritize projects that increase the amount of large woody debris in streams. There used to be a lot more logs and trees and other debris in streams during the, basically the 1900s. It was thought that it was important to clean streams after logging, so fish could get through the streams to spawn. And now we know that large woody debris is really important for habitat. So that's one of the projects that we've prioritized. And also the fish passage work. We have worked to eliminate all barriers in priority stream corridors so fish can get all the way up to the headwaters to spawn and find cold water refuge. And this map just shows that this same area that we've been talking about, Ferguson Creek and the Bear Creek watershed. The green dots are fish passage projects, and the orange stream segments are areas where we've planted trees and shrubs along the creek, and the black stream segments are areas where we've improved in-stream habitat with large woody debris. And you can see Fern Ridge Lake here at the bottom of the map, and Fern Ridge Dam, that pink square, and then the 3 Lowhead dams that I mentioned before, Monroe Drop Structure, the Strota Drop Structure, and Ferguson Drop Structure. So the tree planting work that we've done, we've planted trees and shrubs along more than 30 stream miles in the watershed, 20 of those in the Bear and Ferguson Creek subwatersheds. We've planted over 980,000 trees and shrubs in the history of the Watershed Council, and we're excited to plant our 1,000th tree this winter. Here's some photos of awesome contractors that we use to do that work out there, working so hard in miserable, cold, wet conditions, getting up to 1,000 or more trees planted from each person in a day. This is just a photo or a comparison of 2 air photos of one of our planting projects on Ferguson Creek. You can see in the image on the left, this is before the project. The pasture went all the way down to the edge of the creek where there was a narrow buffer of trees and shrubs. We worked with the landowner to plant trees and shrubs on, I think this one was maybe 8 or 10 acres. This is just one portion of the property. You can see 10 years after the project, visible on Google Earth, you can see that those trees are all established now and helping to not only help shade the creek, but also provide riparian habitat for many fish and wildlife species. The in-stream enhancement projects typically involve placing logs in the stream. We've done that along more than 20 stream miles, 15 in Bear and Ferguson. We've put more than 800 logs back in the streams. We try to leave root wads attached to the trees to kind of mimic natural log jams. Native juvenile fish like to hide in the root, the fine root structure, and the root wads also help with their stability so that log jams stay put as well. Fish passage project wise, this is just kind of a close-up of the Ferguson and Bear Creek subwatersheds again. The green pins are completed fish passage projects, and these orange ones are projects that are underway. I think we actually did this orange one last summer, so we've done a lot of work to improve fish passage. 45 barriers have been removed. We've got 6 more in progress, and almost 30 of those are in the Bear and Ferguson Creek subwatersheds. I think it's really neat that we've accomplished the goal of knocking out all of the priority barriers in a couple of these streams. Owens Creek, a tributary to Bear, and then in main stem Ferguson Creek, we've removed all of the priority barriers and opened up 30 miles of habitat for migratory fish. This is an example of a culvert project. This is the same image that I showed you before. 6-foot drop at the outlet. Impossible for fish to ascend that during this level of flow. What we did with this landowner, this was on private property on Owens Creek, hired a contractor to remove the old culvert with an excavator. We first dewatered the creek, routed around the work area, so we're not impacting downstream water quality while we're digging in the stream bed. Replaced the culvert with a bridge so the landowner could get across Owens Creek, and then also added a lot of river rock to make up that 6-foot elevation difference. So moving on from those tributary projects, our next big priority for aquatic habitat restoration in the Long Tom watershed involves the lower Long Tom River, those 3 low-head dams that I mentioned in Monroe, also upstream on the Strota, at the Strota drop structure, and then at the Ferguson drop structure. This map on the left, it's 2 kind of consecutive panels. If you start on the bottom left here at Fern Ridge Dam, you work your way downstream, and then you jump over to the next panel and keep working your way downstream until the Long Tom hits the Willamette. The blue lines are the current channels of the Long Tom River, and the red segments are historic channel meanders of the Long Tom. After Fern Ridge Dam was built in the 40s, it did not reduce flooding sufficiently enough along the Long Tom. So the Army Corps of Engineers channelized the Long Tom River. They reduced the length from 36 to 23 miles by cutting off a lot of those meanders and isolated them from the main channel. As part of that, they installed those 3 low-head dams to help stabilize that new shortened channel. The first drop structure that a migrating fish hits working their way up from the Willamette River is in downtown Monroe. So this has been a major priority of work for us, working with the city of Monroe, the Army Corps of Engineers, and the Confederated Tribes of Siletz Indians to explore options for improving fish passage at the site. We've been working on this for quite heavily for about the last 8 years, and it's been on our radar for even longer than that. But it's a priority because it's the downstream most barrier, and we've only sampled fish below it a handful of times. But every time we do in the winter, we collect juvenile spring Chinook. So these are a listed species. They're listed as threatened under the Endangered Species Act. So they're a major priority for improving and reconnecting habitat for them. And like I mentioned, these are fish that are born in the McKenzie or Middle Fork Willamette, and then as they grow, they migrate downstream in the Willamette, and then hook a left in west side tributaries like the Long Tom in search of rearing habitat. So we've been working in Monroe for a number of years to explore options for improving habitat for them. We're to the point where we are nearing construction of a dam removal project there. That'll happen hopefully in summer 26. The city of Monroe is the official sponsor of the project, working closely with the Army Corps of Engineers. The Watershed Council has been responsible for securing all of the funds that are necessary on the city's behalf. We're working our way through the fundraising, and we're almost totally done. Project will not only involve the dam removal, but we're also going to have to replace the city's municipal water intake because it wouldn't function once the dam's gone. So it's a big project, and it's super exciting for us, and be a major milestone if we can get it done next summer. This is a kind of a conceptual view of what it might look like in downtown Monroe once the dam is removed. There's a pedestrian bridge that the community has identified as something that they're looking to make happen. So people can access city park property on the left side of the river there from the downtown kind of commercial part of the city. This is just a conceptual image. I think that in actuality, the pedestrian bridge will be located a little bit further downstream, but An awesome volunteer in Monroe made this for us and kind of helped community members visualize what it might look like once the dam is removed. And then after that, there's 2 more drop structures, like I mentioned. So we're working with the Army Corps to start the kind of early work to develop alternatives for fish passage at those 2 next drop structures. And in concert with that, we're working with the Confederated Tribes of Siletz Indians on a Army Corps funded study that will start looking at algae and temperature issues in the Long Tom in more depth, start developing a plan to resolve them, and also do surveys for Pacific lamprey, Northwest pond turtles, and freshwater mussels. I think that's my last slide. So stop the share.
Question.
Hey, does anybody have questions?
Yeah.
What happened?
I was
Anybody?
wondering how much beaver, how much beaver activity there is in the Long Tom.
Ooh, well, we actually might have a public meeting soon. You can put a plug in. We have not seen the results yet, but there is a cool modeling effort done that shows kind of how many beaver probably will be on the landscape in different parts of the watershed. So we'll be presenting that data, I think this winter. We'll have a speaker coming and talking about how many beaver they're actually seeing, at least in similar places in other watersheds. I don't think we've done there have been any actual confirmation or validation of the data about how many beaver you might expect to see in our watershed. And we'll also focus on, I think, different ways people can live with beaver presence on their land.
Right.
So don't have a good answer now, but we'll soon be bringing in some experts to give a presentation on that. But there are a lot of beaver around.
Yeah, I would say anecdotally, like specifically in these 2 areas that we're talking about, Bear and Ferguson Creek, each one of those subwatersheds has like a really large kind of beaver pond wetland colony complex of, like 15 to 25 acres. You can see them from some of the county roads. I think High Pass Road, you can see a big willow beaver swamp up west of where Lavelle Road hits it. And similarly on Grimes Road, which is a kind of a road that comes into Ferguson Creek or Ferguson Road. There's another one of those big colonies that are really neat, just a, kind of a natural wetland area, awesome habitat, and neat to see. And I think anecdotally, landowners, I feel like beavers move up and down the streams looking for new areas to potentially colonize, and sometimes pop up on the floodplain and chew down trees that we've planted or fruit trees that landowners have planted close to the creek. So they're definitely around.
Yeah, I think a lot of our watershed is just like historically prime beaver habitat because they're relatively low gradient streams coming off of like the Coast Range, not super powerful, super big streams. And historically, there was the vegetation there to support them. But yeah, vegetation is a limiting factor now. Is there like enough wood around the streams for beavers, and are people letting them stay is another limiting factor.
Thank you.
Is it, excuse me, is there any softening in pushback about, beavers and their ecosystem engineering amongst the farmers and timber people? Is it still kind of a controversial? I mean, or is it? How do you feel the temperature on them, on that aspect?
Yeah, I'd say maybe there's a slight increase in acceptance of beavers, but yeah, I'm sure that there's still a lot of perceived negative impacts from the beavers on either agriculture or forestry activities close to streams. I know that the state, you guys may be more familiar with some of these rules than I am, but I believe that the state started requiring permits to lethally remove beavers a few years ago. So I would hope that all we do is voluntary work, but I would hope that there are maybe some other regulatory channels that can help people learn to live with beavers because they are so important for habitat and water quality.
Yeah, I have a question related to beaver habitat and wetlands. You had a great map on your website that showed different soil types and hydric soils, which are indicative of previous wetlands. And I'm wondering if there is any increase happening in wetlands in the Long Tom watershed with attempts to restore some of that prior wetland habitat.
Yeah, definitely. Yeah, as you know, yeah, there's lots of historic like wet prairie habitat around like west of Eugene and up north through Junction City, and super imperiled habitat type. It's like down to like 1% of its former extent because it's located on, the prime farmland and close to the urban areas. So yeah, lots of partners work to restore that wet prairie habitat on the hydric soils. The city of Eugene does a lot of good work for that. BLM works on that in the West Eugene wetlands.
Yeah.
We've partnered on a few of those projects. A lot of times it involves restoring hydrology to those wetlands, like plugging ditches or creating shallow scrapes and pools that can hold seasonal water that were probably filled during previous alterations. So yeah, definitely a major priority that we didn't really get to cover much today, but we've been a part of a few of those projects.
Yeah, we'll be helping bust up part of a tile drain network on project soon. So a lot of agricultural land, they installed drainage systems like subsurface drainage systems to help make wetlands not wetlands anymore, make them more farmable. And a lot of those systems aren't well recorded and aren't really visible from the surface. But if we know if they're there, we can deconstruct them as part of restoration, or at least partially deconstruct them to help restore more water on the surface.
Another question, which sort of touches on that is sort of channelization and sort of forcing streams in certain pathways. Is there much work on sort of allowing rivers to flood, sort of to soften the downstream impacts of flooding? Does that, I don't know that you mentioned too much about that type. I know in some other rivers I know of, they've done quite a bit of work to allow overspill at high flood times. Is that a practice or?
Yeah, definitely. I think there's been some amazing projects done, like up the Mackenzie, up by Finn Rock, and up on the South Fork Mackenzie. You might be referring to some of those projects where they've done what's called like it's process-based restoration, where it's stage 0 or stage 8 restoration designs, where they basically fill a channelized river channel and let the river just kind of meander across the historic floodplain and create its own, river adjacent wetlands and log jams and gravel bars and things like that. And I think it's a little bit tougher to do that in the Long Tom. We do have some smaller examples of that we're developing designs for. But because we don't have large areas of public ownership, like the Forest Service in the Mackenzie or in the Siuslaw, and a lot more private ownership and smaller parcels and infrastructure, heavily built along streams that have been channelized, it's harder for us to find those opportunities. But they're, really kind of like the cutting edge in terms of stream restoration. And we are doing our best to work with landowners that are open to those types of ideas to develop those projects.
Yeah, I'll say that like a version of that that's been around for a lot longer is either creating a meandering stream channel or a more natural stream channel, or allowing a stream to reoccupy a former channel. And we've been able to do a little more of that kind of work. It's like a more constrained form of that restoration, where we've been able to let portions of a stream reoccupy a historic channel, or on the lower Long Tom. We are not able currently under Army Corps rules, to move the main channel out of that straight and engineered channel. But we're working pretty hard to find places where we can increase flow and reconnect portions of the historic channel. So it flows both going through the new, like, straight, formerly trapezoidal Army Corps constructed channel and the historic meandering channel at higher flows. So not completely diverting water out of the constructed channel, but still opening up that habitat at higher flows. So doing a good bit of that on the Long Tom.
Great. Any other questions? Another one.
Yep, I'll go on. I'll have another. I don't want to, let's see, what was it? Uh-oh. It was very important, but it'll come back.
While you're trying to think of it, I have one last one.
Okay.
I have many, but
Oh, I know what it was.
one. What towns in the Long Tom watershed depend on the Long Tom directly for their drinking water supply? Monroe, you mentioned. And like, how about Notai, Elmira, other towns?
The city of Monroe is the only municipal surface water source that pulls from the Long Tom. There are groundwater sources that probably like interact with surface water. And then further downstream on the Willamette, of course, there are other municipalities that are pulling from Willamette water that includes Long Tom water from upstream. But yeah, the city of Monroe is the only municipal provider.
Yeah, thanks.
I was just going to ask about, of the three things you're monitoring, E. coli, water temperature, and pesticides, is which of the three, and are there others that you're not monitoring that are actually most problematic? I mean, I look at that, maybe pesticides, farmers maybe want to be more efficient and they'll cut back and be more efficient, and IPM strategies come in. Urban may hopefully improve, but like, what about, I might wonder, is water temperature maybe a bigger threat than pesticides?
Yeah, so it's a good thought. I think it's hard to say conclusively, but especially with warming temperatures globally, with our warming climate locally, that's a factor that we won't have control over locally. Kind of like ambient long wave radiation inputs into our streams, and we know that's getting worse in the future. And there's a limited amount that we can accomplish with shading. There's a limited amount of work we can do to kind of counteract that change. And temperature does contribute to other water quality impairments, including those algal blooms that then contribute to further water quality impairments like dissolved oxygen, fish kills, things like that. So temperature certainly is a really concerning factor, and one that's really critical, at least for native fish downstream. So it kind of depends on like what your lens is when you're thinking about what's the worst water quality impairment. But I mean, that's definitely a big one, and something that people are thinking about a lot is not necessarily how to like cool the entire system to where it was, but how do we preserve cold water refugia? How are we making sure that there's still places that fish can hide out when temperatures get too hot elsewhere? And part of that is restoring fish passages to places where it's colder. Like parts of the map that I didn't really show in the recent temperature monitoring are much cooler. The higher parts of our watershed generally stay cool enough for our native fish. So making sure fish can get up there, and then learning more about cold water refugia, like further down on the Willamette, and how we can protect and enhance those places are going to be really important, I think, in the future, at least from a fish lens.
I mean, I'm in Coyote Creek, feeding into, so I'm in the Long Tom Watershed, and I do contribute. I do throw in some pennies every now. We do contribute, but we're beyond that dam, and so maybe or more of your work seemed urban based. The stormwater, the education through urban, and the focus in lower down. So we're sort of
Yeah.
And it's I think we didn't really talk much about some of the other work that we've done. We are active in different parts of the Coyote Creek watershed as well. So if we had like 2 hours, we could have maybe talked through everything that we're up to. So that's right. I put another plug in for going to our website and looking at that projects map, and just if you ever have questions, just reach out. Contact info's on the website, and yeah, we're getting rolling on some more exciting kind of public meeting topics. We're going to have our annual celebration is in September. We're going to have a tour of our work at Snagboat Bend, which is a large U.S. Fish and Wildlife Service wildlife refuge by the small town of Peoria. We're going to do a tour of that in August. So yeah, we have lots of other work happening, and we just didn't quite have time today to go through it all. But yeah, Coyote Creek's awesome, and thank you very much for the support. And reach out anytime with questions.
Yeah, there's exciting work on Coyote and Spencer Creeks. I think that's Mackenzie River Trust has prioritized that part of the watershed, and we put a lot of effort in there as well, but didn't talk about it as much today.
Well, thank you.
Nice. Yes, thank you very
Yeah.
much.
This has been great. It's 28 minutes after about now, so we should wrap it up. But really appreciate your presentation. A lot of great
Absolutely.
information, and yeah, you have a great website, so a lot of good information.
Great. Thank you.
Thanks
Yeah,
so
very
much. Appreciate it
Thank
so
you,
much.
and thank you for all the wonderful questions and discussion.
You guys are a great audience. Yeah, reach out with any questions via the website.
Thank
Great.
you.
Awesome.
Yeah.
Thank you,
Bye-bye.
everybody, for
Bye-bye.
coming. We appreciate
All
it.
right.
Yeah. Bye-bye.
Bye.
Bye, everyone.